But this number you have to find out that complement. And one more sum. Find 10 complement. 5 to 10 complement. 5 to 10 complement for 5 to 8. It now. So both the sums are choose complement.
Hi students, can you see the screen?
Yeah, both are correct. And this choose complement is choose complement means first find out the one complement for the given number. Okay, one complement you know how to perform. Just replace 0 with 1 and 1 with 0. Okay. So which one?
Hello.
Choose complement. I'll tell you.
Yes, sir.
Can you hear me?
Yes, sir.
Okay. Can you see the screen also?
Yes, sir.
Okay. Nice. Okay. Let me start. So yesterday I have explained you the central dogma of molecular biology, right? So can anyone tell the flow chart of central dogma?
So DNA to RNA, RNA to protein.
Okay, protein. So if viruses are having RNA as a genetic material, for example, if you take the coronavirus, so it doesn't have a DNA, it has only RNA. So if that is the case, how this central dogma will work?
Reverse transcription will take place.
Reverse transcription will take place, and from RNA, what will be formed?
Complementary DNA.
Okay, from complementary DNA undergoes transcription. Then what is the product after transcription?
RNA.
Yeah, which RNA?
mRNA.
Okay, so basically, can you just tell me a single difference between the DNA and mRNA?
Sir, in DNA we have thymine and in RNA we have uracil.
In RNA, what you have?
Uracil.
Okay, that's fine. Uracil, good. And one more? How about the sugar? Sugar moiety present in the DNA and
One is
RNA.
ribose and the other one is deoxyribose.
Yeah. And one more important thing is DNA is very long and it contains two important sequences. Those are called introns and exons. And RNA will not have any introns. It has only exons. So exons are the one that will make a protein, I mean polypeptide chain through translation. So basically that is the central dogma. So now, so now you see you have three important molecules for life: DNA, RNA, and protein. So if, huh? Go ahead. Okay. So nowadays we know that DNA is the genetic material, right? But you just think about 100 years back. I mean, 100 years back, people even don't know what is DNA also and what is the structure of it. But still they used to.
Sir.
But still they used to work with.
Excuse me, sir.
Huh?
Sir, there is a lot of disturbance in your background.
It's a lot of disturbance.
Background, sir. We can hear other noises.
Oh, okay, okay. Yeah, that's true. We, okay, can you hear me now?
Yes, sir.
Yeah. So basically, it was not known 100 years back DNA is the genetic material. So as you have three important molecules and central dogma, protein, RNA, and DNA, and scientists have thought that all these three are important and all these three act as a genetic material. That was 100 years back. And some scientists, they started digging, cannot be a three act as a genetic material, probably one or two. Importantly, RNA or DNA may act as a genetic material. So people started doing experiments, and a lot of experiments were conducted. And finally, there was one scientist named Frederick Griffith. In 1928, he discovered that DNA is the genetic material. So how he discovered this DNA as a genetic material? Because he did a very nice and good experiment with mouse and bacteria. So he took the mouse and he took Streptococcus pneumonia. So basically, this Streptococcus pneumonia that causes pneumonia in humans. It's a small bacterium. So if anybody is having pneumonia, it is because of this bacteria. But if you inject this Streptococcus pneumonia into the mice, they immediately die. Not immediately, they replicate in the mouse body and eventually they die because they cannot tolerate these bacteria, mice. But we also die if we don't use proper medication, right? For mice, I mean, nobody gives medication, right? So as a humans, once we affected with pneumonia, we have to take antibiotics. Then it will clear the bacteria from our body. So basically, there were so many strains of Streptococcus pneumonia, and one of two important strains he has described and he used for the experiments. So basically, the bacteria will have a capsule on its top and a genetic inside there is a nucleus and cytoplasm like any other cell. So basically, the capsule is composed of polysaccharides, I mean carbohydrates. So he took the two different strains of Streptococcus pneumonia, one that causes pneumonia and one that will not causes pneumonia, because the Streptococcus pneumonia that will not cause pneumonia will not have a capsule. So he named them two strains. The one that is causing Streptococcus pneumonia is called type S. Why type S? Type S means smooth, because when he has taken the Streptococcus pneumonia and grow on the agar medium, it has a smooth particles on it and it keeps dividing. And the mutant version that will not have any polysaccharide capsule, he named as a R, rough Streptococcus pneumonia. It is a non-virulent. So basically, you have to know that type S is a virulent and type R is a non-virulent. So in other words, type S will cause pneumonia and type R will not cause pneumonia. Like E. coli, there are several strains. For example, in our body, when you take, we have a small intestine, and when we take the food, in the gut, huge millions of bacteria is present in our gut, because they are useful for digestion of lot of food. They are a symbiotic, which means that they take food from us and they help us digesting the food. So it's a symbiotic. We help bacteria and bacteria will help us. So that is a symbiotic bacteria. So in other words, a pathogenic bacteria also in our body, but if pathogenic bacteria enters into our body, our immune system will immediately activate and it will clear. So pathogenic bacteria, what it does is, it go and conquer the cells and destroy the cells. So basically they are a harm to the human body, and we have an immune system to tackle it. So now Griffith has done a very simple experiment. So he has taken a rough strain, non-virulent, and injected into the mice, and as expected, they are alive. The second line, he has taken smooth strain, that is a virulent, then he injected into the mice. After some days, the mouse dies, because these are the virulent that causes pneumonia to the mice. And the third experiment he has done is, he has taken the virulent strain, he boiled it for a long time, then he injected into the mouse. Surprisingly, the mouse was alive, which means that he completely killed the bacteria and then he injected. So basically these are not useful, they are just particles after boiling. So when they went into the mouse immune system clear them easily, as they are not threat to the mouse, because everything is damaged here almost. So then what he has done is, he has taken a rough strain, which is a non-virulent, then heat killed smooth strain. Heat killed smooth strain means virulent boiled strain. Then he mixed it. Then he injected into the mouse. Basically, the mouse was died. So then he got surprised. He injected non-virulent and heat killed, and how come the mouse dies? Because in previous experiments, he has shown that first lane, they are alive, heat shock, they are alive. But when he mixed it, they die, which means that he suspected that the DNA in the orange color, I mean heat killed smooth strain, is going into the rough strain and converting this rough strain into smooth strain, because the DNA basically is the one that makes proteins, and the proteins that act in the cells, and they are the one that causes the disease. So now what he has done, he suspected that something is happening between these two mixtures, that he called it as a transformation. Why he called transformation? These non-virulent strains are converting into the virulent strains when he mixed with the heat killed smooth strain. So now what he has done, very simple experiment. He took the heat killed cells. And he added the lipids and sugars, and he also taken the mixture of both rough strain and heat killed cells. Then he added one tube, he added a proteinase. So what is the function of proteinase? Can anyone tell? Can anyone tell what is the function of proteinase in the cell? Okay, it cleaves the proteins, it breaks the proteins, and after breaking the proteins are not going to work. And in another tube, this was actually done by another scientist, Oswald Avery. In another tube, what he has done is he added RNase. The function of RNase is to break the RNA. So basically, it completely breaks the RNA. In another tube, he has taken DNase and a mixture, of course. The function of DNase is to cut the DNA, pieces into DNA. So now what he has done is he took this mixture, then he added R cells. R means rough strain, non-virulent strain. So he added to the medium. Then he waited for some time. Then he saw S cells. He is supposed to see R cells intact, but all these R cells are converted into S cells in the tube when there is no protein in the mixture, heat killed S cells. So basically in this heat killed S cells, we have only DNA here and R cells. So DNA and RNA in this tube has undergone something and converted into S cells. Now here he has added RNase. So basically there is no RNA, but still it has a proteins and DNA. So when he added R cells into the medium, again he still see S cells. So basically all R cells are converted into S cells. So finally this is a very important experiment. When he added DNase to the heat killed S cells, the DNase will go and make pieces of DNA so that there will be no DNA, no functional DNA left in this tube. Then he added R cells to this flask where there is no functional DNA of heat-killed S cells. Then he found there were no S cells, all are R cells. So basically from these three experiments, you can say that the DNA in the heat-killed S cells is taking over the R cells and damaging the R cells DNA and replicating itself inside the cell and making it a virulent. So basically this experiment also shows that DNA is a genetic material that can be transferred into the another cell and taking over the cell's DNA if it is a non-virulent, and then it starts replicating. So basically the virulent DNA will go into the cell and taking over of the whole cell. So that's how they concluded that DNA is the one that acts as a genetic material. So again, there is one more scientist called Hershey Chase. Both are American scientists. In I think it's approximately 1920 or 30. They have done another experiment. Very simple experiments. So they have used a bacteriophage for the experiment. What is the definition of bacteriophage? The definition is very simple. Bacteriophage is nothing but a virus, a small virus that go and infect the bacteria. So similarly, like a lot of bacteria come and infect us, right? In a similar way, the viruses that, of course, viruses also come and infect us. At the same time, viruses go and infect bacteria also. So the viruses that go and infect the bacteria are called bacteriophage. And what is the structure of bacteriophage? It will have a very simple structure. It has a head that contains DNA, and it has a neck and a tail. Of course, all these are the protein sheets, and you have a tail fibers. You have a base plate. It's a very simple structure. Basically, this is a virus. So what it is going to do, this virus, it is going to bind to the bacteria. For what? For its survival. Because viruses cannot survive without a host. They need some host. That's why all the bacteria, all the viruses have some hosts. Like bats, like coronavirus host is bats. Pigs have a lot of viruses, and humans have some kind of viruses, and a lot of horse, donkey. So there are several animal species that carry viruses, but those are not harmful to those particular animals, because their immune system is very active and it will damage. That's why the viruses, they just go and sit and they will not harm the animal system. So basically, this is the virus named T2, that virus that infects the E. coli. So a virus that infects the E. coli is called T2 bacteriophage. So what he has done is, he has taken the bacteriophage and he grown the bacteriophage into the medium that contains a radioactive label phosphorus. You can see here the red one. The red one, he has he has grown this bacteriophage into the sulfur containing radioactive medium. Second, the down one, he took some bacteriophage, then he grow these bacteriophages into the medium that contain phosphorus radio labeled medium. So basically he has two flasks, one flask that have sulfur containing medium, radioactive sulfur containing medium, and one flask he has taken phosphorus, radio labeled phosphorus containing medium. So why he has choose the sulfur and phosphorus? Because the virus will have two main components, right? One is nucleus. Now the nucleus will have a DNA, and the second one proteins that are capsulated and protect the nucleus. So we know that all the proteins are made up of sulfur because amino acids like cysteine, methionine, they have a sulfur. And when it comes to the DNA, phosphorus is a main component for DNA backbone. So basically, the phosphorus containing medium, when he grows the bacteriophage take the phosphorus from the medium and it will use it for the replication of DNA. And at the same time, another flask has a sulfur, right? Then the bacteriophage will take the sulfur and it can use it for protein synthesis. So then he grown these bacteriophages into the medium for some time. Then he got a lot of them. In the next generation, what he mixed the E. coli with the bacteriophage in sulfur containing bacteriophages, and another bacterium that have phosphorus containing bacteriophages. So in sulfur containing tube, there is no radioactivity enters. He took the bacterium, the E. coli. Then he checked whether the DNA of virus will have radioactivity. Of course, it will not have a radioactivity because he grown them in sulfur. The DNA cannot take sulfur, it can only take phosphorus because sulfur cannot be used for anywhere in the DNA synthesis or DNA replication. So in the same time, when he grown the bacteriophage into the phosphorus containing medium, once he infect the bacteriophage to the E. coli, of course, the virus will release the genetic material into the cell and it has a radioactivity as it is expected because the replication of the DNA will take up the phosphorus continuously from the medium and phosphorus, so you can clearly see it. So that's how he found that the genetic material is the one that transmits from one cell to another cell. No. And he has also done a very simple experiment. Then he took these both, then he centrifuged. So centrifuged means which one will go to bottom, heavier or lighter? Can anyone tell? What is present in the bottom when you centrifuge the mixture? Heavier particles or lighter particles? Hello? Hello, can you hear me? Hello? Hello, can you hear me? Yeah. Oh, you can hear, right?
Yes, sir.
So which one will go to bottom if you have a
Heavier particle. and lighter ones will flow.
Yeah. So heavier particles will go to bottom, and lighter particles will be in the supernatant, right? So basically, okay, this picture, which one is heavier and which one is lighter? So this is a bacterium called E. coli, and this is a virus. Which one is heavier and which one is lighter? Can anyone guess which one is lighter and which one is heavier? Okay, it's a bacterium that is a heavy. Of course, it will go and bind to the bottom. So then he found all this radioactivity in the bottom, and in the virus there is no radioactivity because they are in the supernatant, right? So now I will tell a simple thing. So once the virus that infects the bacterium and inject its DNA into the cell, you have a protein capsule outside the bacterium, right? So this is called ghost. So probably this is also a very important question. What is a ghost? I mean, bacteriophage ghost. So you can simply say the virus, the bacteriophage that will not have a DNA is called ghost, bacteriophage ghost. So basically these bacteriophage ghosts will appear on the surface of the E. coli, and their DNA will go into the bacterium cell, and it takes over the E. coli DNA, and it will take the control of it, which I will explain in the bacteriophage lytic cycle and the lysogenic cycle. So basically these are the two experiments that has shown to world that DNA is a genetic material, and it is the one that transfers from one cell to another cell, and it replicates very fast. Okay, so basically this is the today's class, and tomorrow's class I will explain a few more experiments for genetic material, and probably you can just go through it. I will probably ask a few questions about this. Okay. Any questions? Okay. Okay, thank you. And I am trying to upload these PPTs. I'm not able to find it. So once I find it, I will upload in the campus technology. Okay, thank you, and see you tomorrow. Bye.
Thank you, sir.
Sir,
Ah.
sir, starting in when the class started, I joined through postdoc, sir. So then suddenly it got disconnected. Then
Okay.
I again joined. I couldn't join through postdoc. I separately joined through Zoom, sir. So I think my attendance might not be noted. Please see to that, sir.
Okay, sure. I will. Anyway, I am taking manually. So
Okay, sir.
today only Shamul and Shamul, one more person. Who is it?
Only he, sir. Others are present.
Okay, only Shamul is absent, right?
Yes, sir.
Okay. Thank you. Thank you. Bye.